So thank you for coming today. Today I'm going to talk about mass spectrometry-based metabolomics. I believe I know many in the crowd here, but there's a few that I don't. So let me introduce myself. My name is Broeckling. I'm the director of the Ark Center, which is primarily a mass spectrometry focused center within Ark with an emphasis on untargeted profiling, including proteomics, metabolomics, and ionomics. So I'm going to talk today about mass spectrometry-based metabolomics, but I did just want to put the schedule for future talks up so that's all visible. And I will also post this at the end. This is also available on our website. So hopefully this is one of many talks that you participate in over the next 9 months, as we've basically filled the calendar year at one per month. With that, I will go ahead and get started. So I'm going to talk today primarily about mass spectrometry. So I know some of you will have experience, others won't. So this is going to be a relatively general talk, but I'm going to highlight a couple of applications toward the end. But hopefully you can use this as introductory material, and it will prompt questions from you that we can expand your knowledge as you move on. So this is what I intend to cover today. First, we'll talk a little bit about omics and why people use approaches like this. I'll introduce mass spectrometry and its application space that we're talking about today, metabolomics, discussing metabolomics generally, and then the platforms that we use commonly. I will get a little bit into the informatics, as it's a very informatics-heavy omic technology. Highlight some resources that are of particular value if you're interested in learning more, and then hopefully answer some questions as we go through. So we'll start by just briefing everybody again on the biological central dogma. Genes are the template, the blueprint that encodes who we are, but it acts through various chemical entities, including transcripts. So genes are expressed into RNA, which encode proteins, one of the functions of which is to perform metabolic reactions that ultimately encode a phenotype. And in biological systems, we're often viewing the phenotype. That's what we see, but more and more there's interest in characterizing at a molecular level as much as possible what is going on that gives rise to that phenotype. That phenotype is an emergent property of all of these various subsystems. And over the past couple of decades, technology has advanced to the position where we can actually look at all of these various biological levels at an omic scale, that is, at large scale. We can look very broadly at genes, we can look very broadly at transcripts, and with mass spectrometry, we can look very broadly at proteins and metabolites. And this is all sort of wrapped up into a systems view that you will see referred to many times in the literature. Specifically today, we're going to talk about metabolomics, and so let's talk a little bit about metabolism and metabolites. And the question is, why would you study your biological system with metabolomics as opposed to one of the others? And so we're focusing really on metabolism here. And metabolism is a very highly networked, or often very compartmentalized series of biochemical transformations that's used to power life. It's a very generalized description of what metabolism is. But really it serves as the underlying sort of currency on how all biological systems work. And this includes things like energy generation. So when we think about plants converting carbon dioxide into biomass, that is done through metabolic reactions. And those metabolic reactions can be traced through tools like metabolomics. We can think about a nutrient budget. We often have crops, for example, that are limited by nitrogen availability, and that nitrogen budget is governed largely through metabolic processes. If we think about the human system, obviously our metabolism is impacted by what we eat. And you can think of the ingestion of vitamin C as a way to prevent scurvy as one of these metabolic reactions. This actually involves both metabolic transformation as well as proteins, as it's often collagen that is the impact that vitamin C has. It's on collagen structure to prevent scurvy from developing. Of course, there's also basic biomass generation and maintenance. So all of the stuff that we're built of, a lot of that is polymers. All of the things that those polymers are built of are incorporated into basic metabolic pathways. So metabolism fuels all of the other OMs as well. And then finally, there's a large interest in metabolomic technologies in the clinical world because many human and animal diseases actually have a pretty strong metabolic component, including things like cancer, obviously obesity. So these small molecule metabolites are often pursued with respect to biomarkers, as well as fundamentally understanding the metabolic processes that give rise to a given phenotype, which may involve disease. So metabolomics as an approach really started to emerge around the year 2000, and there were a couple of papers that came out around that time, particularly using gas chromatography mass spectrometry to profile broadly. And this was a combination of some derivatization chemistry, people starting to recognize that when they did their targeted analyses, there were a lot of other signals that were present. And then finally, this paper, Metabolite Profiling for Plant Functional Genomics, an author, Oliver Fien, and the group that I was working with, really sort of put this together to realize that you could be applying these analytical techniques in a manner similar to that being used to profile transcripts or genes. So it was a move from targeted analytical focused technologies, adapting existing technologies in a new, more omic centric manner. So it really was sort of a step change. It was a gradual change, but the implications of it became more and more apparent as it was sold in this context, plant functional genomics. So what is metabolomics then? The metabolomics really is an analytical technique which has an aim to qualitatively and semi-quantitatively profile all of the small molecules in a sample. This is really an unattainable goal, and I'll give you some examples as to why that's the case. But this is ultimately the big picture function of what metabolomics is designed to do. Now, I use the term metabolomics in a very inclusive manner, and I just want to warn you that not everybody does. So as I'm defining the term here, this is basically all molecules with a molecular weight less than, say, 1,500. And that's a round number. You could set it to 1,200 or 2,000. Various people use different cutoff, but we're talking about generally all the small molecules. And this includes things like the small polar metabolites, glycolytic intermediates, gluconeogenesis, Calvin-Benson cycle, TCA, all of these small amino acids that you commonly think of as key components in metabolic processes. But it also includes things like lipids, and some people will refer to lipids in a separate omics, so we'll call it lipidomics. I'm going to be inclusive and use it under the metabolomics umbrella here. But this includes things like membrane lipids, plant waxes, cholesterol. So it's a relatively diverse class of molecules in its own right. This also includes small molecule hormones. We can think about, for example, in mammalian systems, testosterone or estrogen, these small molecules that have signaling function. Now, these are small molecules. They certainly fall under the realm of metabolites, but they're generally at very low concentration, as most signaling molecules are, which can cause their challenges in their measurement. This also includes things like natural products or specialized metabolites. You can think about caffeine in coffee beans. Caffeine isn't there for a biomass function. It's not there for basic metabolic maintenance. It's there serving a specialized role. And when we're talking about caffeine in coffee, that's a natural product. Now, we can also think about caffeine in humans. So if I had my morning cup of coffee, that caffeine there is now something I have ingested from the environment that it might be considered an exogenous compound. And this has been applied its own omic name at times called exposomics. But the simple fact is that if I analyze a plasma sample from myself, it's going to include both human metabolites, compounds that I have ingested, lipids, and those hormones. So when we're thinking about this, and the reason I define it inclusively is we have the possibility of seeing analytes from all of these various classes. Now, we do this through tools primarily using chromatographically coupled mass spectrometry. I'm going to spend a little bit of time talking about that technology, why it's used, what platforms are used, the data that is generated, and how that feeds into the metabolomics approach. So why mass spectrometry? There's actually other platforms that can be used for metabolomics, but I'll first talking about the strengths of mass spectrometry as the reason that mass spectrometry has become sort of the default go-to for metabolomic analyses. And really the two big ones are resolution and sensitivity. Chromatographically coupled mass spectrometry offers resolution of a really complex small molecule sample by mass. That is, the mass spectrometer can separate molecules of different masses from each other. But it also enables the coupling to chromatography, direct coupling. And that means that you immediately have two dimensions of separation that you have available to take that really complex sample and make it simpler for your analyses. There is also, over the past decade, the development of ion mobility. This is a relatively newly available technique on some instruments, which can offer a third resolving dimension. We're not going to talk too much about that today, but I wanted to bring it up because it is out there in the literature. And the other side of mass spectrometry that's made it extremely attractive is its sensitivity. This sensitivity means that you can directly couple chromatography to mass spectrometry. It also means that you have relatively small biomass demands. So you don't need a lot of tissue to develop a good, or sample to develop a good metabolomic assay. And this is particularly important because it's sensitive both in an untargeted manner, that is when we're looking at all of the signals, and I'll talk a little bit about untargeted versus targeted in a couple of slides, but it can be extremely sensitive with targeted approaches. So mass spectrometry on the whole is widely adopted for these two general properties, resolution and sensitivity. NMR is probably the most frequent alternative, so I wanted to bring it up here. Generally speaking, it is less sensitive. Generally speaking, it is less resolving, though it does have the ability to actually resolve individual atoms in molecules, which can be of value at times. It does have some strengths. One of its strengths is stability. When you introduce a molecule into a mass spectrometer, it doesn't come out. What that means is you actually get your mass spectrometer a little bit dirtier every time you run an injection. With NMR, that's not the case. The molecules that you put into an NMR in your tube come out in your tube, and therefore the NMR tends to be a much more stable platform. And it is also more inherently quantitative. So I don't want to discount the value of NMR, just want to highlight mass spectrometry as a frequently used tool for this application. So let's think a little bit about what a mass spectrometer is right now. As I'm describing it, we've got chromatography coupled to mass spectrometry. Now, chromatography in our case could be liquid chromatography or gas chromatography. In this case, it's irrelevant. We can just think about chromatographic separation on the front end. Our second step, the first thing that happens once molecules come out of the column is we need to ionize them. And in this case, I've mentioned electrospray ionization, but this could also be electron impact ionization or chemical ionization. And this turns the molecules coming out of the column into ions. After ionization, we have typically a quadrupole type mass selector, which is a mass filter that we can scan through all the masses or just selected masses. We often have what's called a collision cell, where we can take an ion isolated by the quadrupole and fragment it to generate an MS-MS spectrum. And then we have some sort of mass measurement device. The two most commonly used in untargeted metabolomics are a time-of-flight mass spectrometer or an Orbitrap mass spectrometer. There's a lot of different instruments out there and a lot of different instrument architectures. This is a generalized one, which is generally applicable. And I threw ion mobility in there. You'll see ion mobility referred to in the literature. They're in different locations in different instruments. So I drew arrows to a couple of different locations. But the bottom line here is that it's good for you to have an understanding that what we're doing is separating with the chromatography, ionizing in an ionization source, using some sort of combination of mass selection devices, and then detecting the ions with a detector at the end of the flow path of the ions. And so at the end of all of this, you have resolution by chromatography, resolution by mass, and signal intensity from the detector. And it is this trio of resolving dimensions that we can use to detect and identify our molecules of interest. Now, broadly speaking, there's two approaches to metabolomics, untargeted and targeted. And I'll just briefly describe what those refer to so that you're aware of them. Most of what I talk about is going to be untargeted. We will have another talk later in the series on more targeted approaches. So generally speaking, an untargeted metabolomics approach uses very generalized sample prep. You have an idea of what instruments you want to collect your data on. You know what types of molecules you're expecting to see. You can use a relatively generalized method to prepare your sample. Use very generalized data acquisition, that is trying to collect all signals. And generally this is done on a TOF or Orbitrap, though GCMS are also used. Use very general data processing where we're trying to detect all signals. So I've mentioned here the generalness, but also what comes with that is the breadth. So this really has driven the strength of untargeted metabolomics. That is, you have the opportunity to see all of the signals that are present. And that is really valuable if you are looking to learn more than a very targeted focused hypothesis testing. So some people refer to untargeted metabolomics as hypothesis generation, and it certainly is that, but it's not limited to that. You can certainly do hypothesis testing with untargeted metabolomics as well. It's very useful for discovery and very generalized methods, and that comes in very valuable, particularly when you're moving out of really highly characterized model systems, because metabolomics approaches used in human samples are very similar to those used, for example, in maize or in specialized crops or in soils, for that matter. A lot of the chemistry and a lot of the informatic tools are the same. That contrasts with targeted metabolomic approaches, where you used very specific sample preparation methods targeting a particular analyte or analytes, and you want to optimize your methods for that analyte. So we also then use very specific data acquisition methods. Generally, we're using a tandem quadrupole. So rather than a quadrupole collision cell TOF, it's a quadrupole collision cell quadrupole, and that increases our sensitivity. This also means we're using very specific data processing approaches. So the strengths of targeted metabolomic methods include sensitivity. Oftentimes you can have 10-fold higher sensitivity in a targeted assay than a comparable untargeted, and also streamlined data analysis. So they each have their strengths, and different approaches are going to be of value under different situations. Now, metabolomics also brings challenges, and I think it's worth discussing these as you think about metabolomics as a platform that you might be applying to your research. One of the biggest ones is chemical diversity. So HMDB is the Human Metabolome Database, and this is a relatively well-curated database. It has about 253,000 metabolites in it in the current stage as of yesterday, and that's a relatively focused one. PubChem has about 115 million structures in it. Now, many of these are not ever going to be seen, but still it demonstrates the scope of the chemical diversity that we need to consider when we're thinking about metabolomics. So really what it comes down to is that you should understand that one solvent or one extraction doesn't extract all the small molecules, unlike DNA or RNA. And just as importantly, one analytical platform doesn't detect them all. So this chemical diversity generates its own challenges on its own. An additional challenge that we struggle with is quantitative dynamic range. I mentioned the presence of, for example, hormones. So you could imagine, just picture something like, for example, sugarcane, where you have extremely high concentrations of sugar. Well, those hormones that are driving the development of sugarcane are also present at extremely low concentrations. And being able to measure those really, really low abundance things in the presence of a really high background of something else can be a challenge. Another one that we struggle with compared to some of the other omes is it's really the only ome which doesn't directly map to the genome. So if you're doing proteomics, proteins can ultimately be mapped to the genome through their transcripts. That is less easy to do and less easy to do well with metabolites, especially when you're talking about specialized metabolisms of microbes and plants and fungi, where you have these specialized metabolites, which the genes of which aren't really well characterized. That can make annotation challenging. We do struggle at times with instability of the molecules that we're trying to analyze. When you have labile energy metabolites, they're just not stable in solution at room temperature. So you have to treat your sample with some level of caution as you're doing your prep to make sure that you can measure them quantitatively. We also deal with, as I mentioned before, instrumental instability. We're actually introducing sample into the mass spectrometer and it doesn't come out. So what that results in is signal drift in the mass spectrometer that we need to be mindful of and being able to correct for. A lot of the contaminants that we have in our systems are actually small molecules. And some of these small molecule contaminants are actually the same molecular structure as the analytes we're sometimes looking for. So that can be a problem. Contamination is something that we've got to be mindful of. And finally, when we get into the data analysis, there's a many-to-one relationship. And I'll display one of these in a minute. But basically, one molecule doesn't give rise to one signal. So our informatics needs to be mindful of all of these properties. There's primarily two platforms in mass spectrometry that are commonly used. One is gas chromatography and the other is liquid chromatography. I've tried to put up just a quick summarization of the properties of these and how well they perform relative to each other. But the take-home message should be that there are times where gas chromatography is going to be your preferred method, and there are times where liquid chromatography is going to be your preferred method. And it's useful to look at the literature and to see how other people have analyzed similar samples. And it's useful to talk to your local experts and try to see what's available and what might be the best platform for your research interests. But generally speaking, GC is great as long as your compounds are compatible with GC. If you look at the analytical analyte coverage in the table, GC is good, LC is great. And so you can analyze more things on LC, but oftentimes the ones you can analyze by GC, you can do better on GC. That's not universally true, of course, but oftentimes it is. There are other platforms that people do use for metabolomics. This includes flow injection, which is basically LC-MS without the liquid chromatography column. So you take a liquid sample and you infuse it into the mass spectrometer to eliminate the separation. This is much, much faster than liquid chromatography based analyses, but you do lose the chromatographic dimension. So things like isomers are no longer resolved under a flow injection platform. You will also occasionally see capillary electrophoresis coupled to mass spectrometry. And there's some really cool techniques out there called mass spectrometry imaging, which are used to study small molecules or metabolomics as well. We don't have an imaging platform, but I did want to bring it up here because it is exciting and you will see it in the literature. So I wanted to introduce briefly what the data looks like coming out. Now, metabolomics data in its raw form is effectively three-dimensional data. The first two dimensions are the mass to charge ratio, that is the mass dimension, and the intensity, the signal intensity, and this is a mass spectrum. So this is a sort of a theoretical mass spectrum for if we were to analyze cholesterol by electrospray in negative mode, this is what it would look like. And you see there's more than one signal there. We see a signal at about 385.2, another one at 386.2, another one at 387.2, and a tiny, tiny little signal at 388.2. Those represent isotope signals. So this is an isotope envelope. And the reason you see this, rather than just one molecule, one signal, is that there is a certain natural abundance of 13C in all carbon that naturally occurs. It's about 1%. And so if you have a molecule that has around 30 carbons, as cholesterol does, then there's a very good chance that any one of those is going to have 13C instead of 12C, which increases the mass by 1. And so what this effectively is when you're looking at this isotope distribution, is a probability distribution for how many 13Cs are in the molecule. And so we see several signals, and we can actually use this information to help constrain how many carbons might be in the molecule. So when we're getting this information out of a mass spectrum, we have the accurate mass if we're using an appropriate mass spectrometer, and we also have its isotope pattern. And those are both useful in helping to assign structure to an unknown signal. So those are the first two dimensions. The third dimension is retention time, as I had mentioned before. And so this is an example of what raw data would look like in a three-dimensional plot. In this case, our x-axis going left to right is the mass dimension. There's going to be a sort of a y-axis going back into the screen. That's your retention dimension, your chromatography, and then you have intensity going top to bottom in your z dimension. And so all three of these dimensions are useful in separating signals. And we have to use data processing approaches which recognize this inherent data structure to pull out as many of those little green spikes as we can, because they all might represent our new biomarker or a really interesting discovery. So the data processing that we use goes through several steps. The first step is to find the signals. So we take all of the samples in the data set, and in each one we try to find all of those little green spikes. Our second is to align the signals across the data set. So if we see about the same mass and about the same retention time in two separate injections, they probably belong to the same molecule in two different samples. So we want to make sure that those are aligned. We often end up with missing values, so you can report missing values as missing, or you can assign some sort of value to them. And missing value imputation or assignment is an important consideration when you're doing data processing. Next, we want to actually group features. So when I mentioned that we have a many-to-one relationship, we want to make sure that all of those cholesterol isotopes are recognized as such and put back together into a cholesterol isotope envelope, because by default, the initial signal detection doesn't recognize that. We often end up with batch effects because in larger studies, we want to make sure that we account for those batch effects through appropriate experimental design and controls. Normalization serves a similar function. And then finally, we want to do statistical analysis. Now, all of these steps that I described are relatively constant. That is, whether we're analyzing soil or human blood or plant leaf material, those processes all occur in a very similar fashion. What changes are these last few steps. The annotation process is more dependent on what type of sample you have, and it's not so much that the process of annotation is different, but the data that you use in the annotation process should be different. If we are analyzing human samples, it makes sense to use the human metabolome database. If we're analyzing coffee bean, maybe not. We want to try to find a database that is a little bit more tailored to natural products and the analytes that we're likely to see there. And then finally, one of the most challenging steps is actually the interpretation. So once you get all of this metabolite data, what does that mean for you biologically? And so that is something that is obviously extremely dependent on the type of samples that you're using. So I want to bring up the notion of MSMS. You'll see this a lot in the literature, and it's something that we're very dependent on in annotating our unknown signals in untargeted metabolomics. And it's also something we're very dependent on when we're doing targeted metabolomics for increasing our sensitivity and selectivity. So the schematic that I showed earlier, we have our chromatography followed by ionization. We have a quadrupole and a collision cell. MSMS is really the process of taking an ion, using the collision cell to collide it with a noble gas, typically an inert molecule under vacuum, so that the ion of interest breaks and falls apart into a diagnostic MSMS fragmentation pattern. We then measure those product ions, as they're called, with our time of flight or Orbitrap detector, detect them to spit out a schematic blue MSMS spectrum that you see on the right. Now that MSMS spectrum, we have our signal intensity and our mass to charge, and at a given collision energy for a given ion, under given conditions, that's extremely reproducible. So you can take your MSMS spectrum and compare it to a library of MSMS spectra, for example, to identify what that might be. And this is actually very similar to what's done in GC electron impact, where fragmentation is inherently part of the process of ionization, and we use libraries to annotate in that case as well. So what I've described here, that you'll see the term data-dependent acquisition. That's a form of MSMS acquisition where the precursor selection, so if we're trying to measure caffeine, for example, in coffee, we can tell the instrument when you see the mass 195 trigger an MSMS event, and isolate the 195 and fragment it. And that is called data dependency in the MS-MS acquisition process. So the MS-MS event is tied to the MS1 signal. That is in contrast, and you'll see this more in the literature, so I wanted to introduce it here as a concept. We won't get into the details because it's quite complex at times, is data independent acquisition. And this is a form of MS-MS acquisition where you set all of your precursors before acquisition begins. And generally speaking, you're using wider isolation windows to try to isolate all of the ions that you might be interested in all of the time. And that comes with some strengths and weaknesses, which are a little bit beyond the scope of what we want to talk about. But if you ever are interested in learning more about that, don't hesitate to reach out. I want to introduce also the notion of quality control. This is a really important component in metabolomics specifically, but really in any omics. And so some things to be thinking about as you're thinking about a metabolomics experiment are ways to demonstrate at the end of the day that your data is of high quality and is reproducible. And this begins with experimental design, making sure that you remove your covariates, that you have good replication. This includes sample preparation, things like accounting for contaminants that might be introduced through the sample preparation process by including process blanks. So taking a blank vial and treating it for a real sample and actually analyzing it at the end of with your real samples. Randomization is always important. Data acquisition, we also randomize the order, the injection order and analytical order for that. We often have some component of analytical replication. If we inject the same sample multiple times, how consistent are those data? Data processing obviously has to be optimized not to give you the maximum number of things, metabolites in this case, but to make sure that those things are reproducible. Ultimately, your data is best deposited in a repository, and more journals are looking for deposition in a repository as part of the publication process. So just be mindful that that might be part of the expectation as you're going in. And then finally, in the statistical analysis, you've got to be very mindful of the fact that you're looking at thousands of things, and there is some special consideration that you need to be aware of as you go in. So I put a link here. This, again, will all be available online. But Keith Baggerly is an excellent presenter. He's a statistician by training, and he's sort of made a bit of a name for himself in looking at published omics work with a very critical eye. I highly recommend popping in and watching a few of his seminars because they're very engaging and entertaining and also very enlightening. So I just wanted to highlight one of these. So this is an example for the importance of false discovery rate control in omic research. So this is completely a theoretical experiment here. This is completely random data. And let's say we've got 3 replicates of a control and 3 replicates of a treatment, and we detect 2,000 metabolites. If you use a random number generator and you do an ANOVA on those results, this is what you see. So for your 2,000 metabolites, we have about 5% of them which have a p-value of 0.05 or less. That is not a coincidence. We're setting a p-value of 0.05, which is going to guarantee that in random data, we are going to see some significant results. The more tests you have, the more significant results you would expect to see. And I want to emphasize, because I've heard this comment that, well, it might be that you have significant results, but if you just look at the box plots, you can tell which ones don't look real. But this is one of the most significant p-values, and it looks pretty real by a box plot, but it's just a random sampling artifact. So the way that we control for this in an omics setting is to use what's called false discovery rate correction, or FDR correction. And it's a way to adjust the p-values such that post-correction, assuming standard normality constraints in your data, all of those insignificant, falsely significant p-values become insignificant. And so this false discovery rate correction is a really important component, and it keeps you from misinterpreting data based on the fact that you're just sampling a large number of things and doing a lot of tests. So there's a lot of data processing software out there. I rely very heavily on tools within the R environment. But I wanted to highlight not only that, but some of the more popular tools that are out there outside of those. So it's important to look at your raw data when you have the opportunity to do so. And usually the best way to do that is to actually use your instrument's data viewer. So with Waters, that might be MassLynx. Thermo has Freestyle. All of the vendors have their own programs for viewing the raw data. And you can often view those very easily in those raw formats, looking at your blanks, looking at your samples. How different are they? What are the major signals? If you want to get out into some of these open tools, you often have to go through a conversion step. So there's a program called ProteoWizard, which can help get you into those open formats. MZML is the most sort of the standard format that most LCMS and MSMS data goes into. And then there are a lot of processing tools out there. I mentioned R-based tools. XCMS was developed in R initially, which is one of the reasons I first started using it, but there's a lot of other tools within R. And I put a link here to a whole online book that you can use to reference all of the tools available in R that are of value in metabolomics work. MZMine and MSDial are two commonly used graphical user interface type programs, which can also be used for processing metabolomics data. The annotation process in metabolomics is dependent on the type of platform you use, but there's a couple of steps generally. One is inferring the molecular weight. When we're doing LCMS, when we've got soft ionization, we try to infer what the molecular weight of the signal is. This can include looking at different adducts of the same molecule and using those to improve your confidence, for example, looking at the isotope pattern. There's a lot of spectral matching tools out there for when you do have MSMS data to support your annotation. NIST is the National Institutes for Standard and Technology. They've got a large library, including GC electron impact and LC MSMS libraries for annotating unknowns and extremely high quality data. GNPS is Global Natural Product Social Networking tool. This is actually an online platform where you can upload your data to and link it not only to known libraries of authentic standards, but also libraries of all of the other people that have uploaded data. So you might be running, for example, soils, and you might see a signal that you don't know what it is, and you could upload your data to this. And even if there's not a library match, if somebody else has analyzed soils and that signal is present and you see a similar MSMS spectra in that data set, it would match. And so it is sort of this global, extremely large scale spectral similarity network type application that can help link studies across a scale that was otherwise really not feasible. There are also tools for a spectral interpretation, and you could try to interpret your spectrum manually. Obviously, that takes experience and expertise. Tools such as MS Finder and Sirius will take your unknown MSMS spectra when you have accurate mass data and try to, and a library of structures, and try to prioritize which structures are the most likely to have given rise to your MSMS spectra of interest. And I do want to mention that there are other analytical informatic approaches to help you to make sense of your data. So we are dealing with some samples in the lab right now, which have a fair polymer content in them. And so there are tools out there, for example, that perform homologous series detection. And I've got a small plot there, which shows you every one of those little points is a MS feature that was detected. And whenever they're connected by lines, that suggests that it's part of the same series. And this might be things like polyethylene glycol, for example, where you have increasing mass at increasing retention time in a predictable manner. And interpreting one of those then gives you the ability to interpret all 5 of them that are linked in the homologous series. And there's others out there. So annotation is a very big challenge in metabolomics, and there's a lot of tools out there for supporting that. Interpretation can often be challenging. There's one of the most useful tools out there and most accessible is a tool called Metaboanalyst. This is developed by the same group that developed the Human Metabolome database. So this is particularly useful for mammalian samples. That being said, there's a lot of common pathways that are built into it, metabolic pathways that can also be used to help interpret data. And it's a web-based platform. running R in the back end. So you can actually download all of the R packages and use it locally as well, but the web-based is often easier as a first step if you're going to be using it once off. It is fully mapped to the human metabolome database and includes statistical analyses such as univariate statistics, multivariate statistics, pathway analysis, and pathway enrichment type statistics. And more recently, they actually added raw data processing functionality into it as well. So there is functionality. I haven't used it, so I don't know what it's like for taking raw data and going through all of those steps that I talked about prior for processing the data as well. And I will also mention that there's in the R environment, there are a lot of packages out there for improving interpretation of your metabolomic data. So I just wanted to highlight here, there are several systems that are present in Ark that are available for use, including several GC systems, including GC electron impact and chemical ionization, as well as atmospheric pressure chemical ionization. We do have one GC, which is connected to a time of flight for accurate mass measurements. Otherwise, we have quadrupole and tandem quadrupole instruments, which can be very useful for metabolome analyses. And we also have several LC systems that are of value here, including a Bruker QTOF, Agilent QTOF, and Two Waters QTOF, one of which does have the ability to perform ion mobility spectrometry as well. And so we do have a lot of resources here on the instrumental side to enable metabolomics experimentation. You can always look on our website as well. So under our website, under our equipment, you can find a searchable table, and you can search for mass spectrometry and see a full list of all of the mass spectrometers available. One of the instruments, for example, that I didn't mention as part of our metabolomics, we do have an LC Orbitrap system, for example, which is commonly used for metabolomic analyses. We use it primarily for proteomics. But you can see all of the instruments in their current status by going to the website. And I also want to highlight as resources all of the people within Ark that can help with this too. Ultimately, there's a lot of expertise that goes into not just metabolomics, but mass spectrometry generally. Claudia runs a lot of the self-service mass spectrometry. Linsing runs a lot of our targeted metabolomics. I run a lot of our untargeted, working with Nathan Montgomery over on the right. Caroline and Paul work a lot with GCMS over in the chemistry building. So there's a lot of human resources here that you can call upon when you have questions as well. So I just really quickly, in the last few minutes, wanted to highlight a few applications that we have worked on in the past year, for example. We'll start with Jacob van der Roost and Thomas Bork in soil and crop sciences. So this is a project where Jacob is a student, and he was interested in looking at the impact of fire, forest fire, on soil microbial communities, and in particular, interested in understanding what these small molecules are that are produced by fire or released by fire that might be suitable for fueling microbial growth. And so we actually put together this pyrocosm experiment, which is drawn in schematic here. And basically the goal of this experiment was to then create fires under controlled conditions, recover metabolites that were released during burning and into the soil, and then measure those metabolites. In this case, we chose to use GCMS, given the interest in small polar metabolites, water-soluble things. And here's an example of a principal component plot looking at burned in red on the left and unburned samples on the right. And you can see large differences in their profiles. Effectively, this is a multivariate tool looking at the metabolic profile of these soils in response to burning. And in the middle there, this is just to highlight some of the quality control that we can build in. If we take a sample that is sort of an average of all of those by taking averages literally of the extracts and running them with the sample set, we can see that our quality control samples sit in the middle where we would expect them to be when data quality is good. This is a way to demonstrate not only that we've got good data that makes sense given the experimental design, but that it makes analytical sense and that we have good reproducibility. Another example, this one from Horticulture and Landscape Architecture, Melanie Odenkirk and Jessica Prenny, are part of a project called PTFI, Periodic Table of Foods Initiative. This is a large initiative funded by Rockefeller with the interest of actually developing very standardized methods for food analysis using liquid chromatography, accurate mass spectrometry. And so basically the goal here is to be able to take any food and in any country, in any laboratory, perform an analysis under very standardized, controlled, standard operating procedures so that the data coming out can all be piped into a given data pipeline, data analysis pipeline, and deposited into a public repository that is available. So this is a really cool project on many levels, but one of the coolest and where I think it's of particular interest to the metabolomics community is it's probably the most standardized effort at untargeted data acquisition anywhere, as far as I'm aware. And that includes, in comparison to, say, untargeted clinical analyses. Very, very high degree of rigor going into this in an attempt to standardize. And there's one more. This is a recently published paper from Giovanna Catandi and Elaine Carnival in Department of Biomedical Sciences. And their goal was to try to actually look at the impact of these nutritional supplements on reproductive health. And so they used, in this case, largely a lipid-focused method using LC time-of-flight mass spectrometry to look at the lipid composition in these oocytes and blastocytes and see how the nutritional supplements impacted those profiles, and then actually correlated it to mitochondrial health. And so these are all published data, in part with data from our group using our LC time-of-flight instrumentation. And so the different applications, all using very similar technology. I wanted to highlight before we end a few additional resources. There's a lot of societies out there that you can learn a lot more about this, including the Metabolomics Society of North America, an International Metabolomics Society. The meeting I go to every year is the American Society for Mass Spectrometry, and we have a local mass spectrometry society group as well, where we try to get together a few times a year and talk not just metabolomics, but biological mass spec more generally. And of course, you can always find more information on our own website and reaching out to the people listed there, myself included. And with that, I would like to open it up to questions.